Morning, everyone. I hope you guys got to enjoy a bit of a late start, get outside, enjoy the nice weather on your way over here. We're going to try and get started. We have Dr. Barb Milosky and Carmela Pepe, who are going to be co-moderating this first session, and I'll get out of the way so that they can introduce our speakers.
Good morning, everyone. Welcome. So I have the pleasure to introduce the first speaker, who is Dr. Patrick Ford. He couldn't be with us this morning, so he's going to give his presentation remotely. Dr. Ford is a medical oncologist from Johns Hopkins University in Baltimore, and he'll be speaking about how to explore the rational IO combinations in early stage disease. So we will connect with Dr. Ford.
Thanks very much. I'm sorry I couldn't make it in person. Fortunately, I was struck down with a fever a few days ago, but feeling better now. I'm going to just share my screen. Can you see that okay? It's in presenter mode on my side. Is it the same on that side? Yeah, that's good. Okay. So I think John and Normand had asked me to discuss exploring rational immunotherapy combinations in early stage disease. So it'd be great to have an answer to this question, but it poses more questions than it does provide answers at the moment. These are some of my disclosures. So you've probably heard all of this over the last couple of days, but this is kind of where we stand at the moment, at least in the US and in several countries internationally, in terms of approvals for early stage non-small cell lung cancer. The first approval obviously was in PAR-010 in the adjuvant setting, and most of that benefit in PD-L1 high. Then the Checkmate 816 study and Keynote 091 shortly successively, and more recently the Keynote 671, the first perioperative trial to be approved and to show an overall survival benefit. There's some things in common here. All of these regimens in various components require patients to be eligible for chemotherapy, and they don't really take account of clinical or pathologic stage, whereas in the neoadjuvant setting, we're talking about clinical stage, which is likely higher risk and probably less well-defined. So I'm going to talk a little bit now about the history here. So when I started as a fellow back in 2010 at Hopkins, it was the first phase 1 trial of nivolumab had just been published, and successively over the following 4 years, there were multiple advances. So it looked like this couldn't be beaten, leading up to 2015 when nivolumab had a positive result in the second-line setting in non-small cell lung cancer. And this was the first real advance in this setting in many, many years. And the perception among many people, and myself included, was that there was a strong movement away from chemotherapy. And there were many media presentations about this at the time. But 9 years later, we haven't changed a whole lot. There's been, so I was counting them over the last few days, about 22 different negative large phase 2 or 3 trials of immunotherapy combinations. The only approvals we have that are immuno-oncology for non-small cell lung cancer are PD-1 or PD-L1 and CTLA-4. And as you can see, it's more common to see these press releases of negative trials than it is of positive trials. I was involved in some efforts kind of in the earlier days, shortly after that 2015 approval, to develop more rationally combinations of immunotherapy or immune-based therapies. So this was the FRACTION trial, which was quite novel kind of at its time. It was conducted from about 2015 to 2017, involved combining, having a control arm of nivolumab, and then combining nivolumab with multiple novel agents, both in patients who had not been exposed to immunotherapy and in patients who had prior immunotherapy. Unfortunately, what we found that most of these arms did not show significant benefit. There have been advances on this. So the Hudson trial, also an advanced non-small cell lung cancer, similar patient population here, focusing purely on patients who had prior immunotherapy. So patients who had at least one line of immunotherapy and one line of platinum doublet chemotherapy, Patients were excluded if they had EGFR or ALK or other targetable alterations. The key difference here is this was separated into a matched population where the patient had a particular marker which might predict benefit from the agent, and a non-matched population. And I think this is key because sometimes when we focus only on the matched population, we don't really know if that is truly just an enriched population or if it's only the population who will benefit. And we're seeing this a little bit with ADCs, where trastuzumab deruxtecan in breast cancer has migrated from HER2 positive now to HER2 low and perhaps even HER2 negative. Whereas if we had explored it in a design like this initially, perhaps you would know those data up front. But in any case, this was a very large study. So Elsa Ignacio, who's in the audience, I think, or at the meeting, is very involved in some of the translational science here. You can see from the CONSORT diagram, it became complicated. It involved a lot of pre-screening. So these patients all had a pre-treatment biopsy. That biopsy was sent for clinical NGS with Foundation Medicine. And based on those findings, along with some IHC tests, they were then randomized on the study either into a matched cohort or a non-matched cohort. So what were the results? Well, of all those arms, one of them graduated, and that was the durvalumab ceralasertib arm, which was an ATR inhibitor. And you can see here, the primary endpoints of Hudson were somewhat of a mixture of response rate, PFS, and OS. And in all of these metrics, durvalumab ceralasertib graduated better than the other regimens. So you'll see there was an improvement in PFS with the combination, and also an improvement in OS, relatively speaking, compared to all the other regimens. And LATIFY, which is the phase 3 trial here, has just completed accrual. This is essentially comparing ceralasertib durvalumab with the standard of care docetaxel. So you might argue that compared to most of the trials we do in this setting, so for example, and the recent trials with TROP2, there's probably a lot more data coming from Hudson to support LATIFY than there was, say, for example, to support some of the ADC trials we have seen, or some of the combinations like the VEGF TKI plus PD-1 in that setting. So hopefully this trial has a higher chance of success because of that. And the reason I go through all this is that perhaps this is one model that we can use to inform development of rational combinations in early stage disease. We can also learn from other tumor types. So the I-SPY2 trial, which is an adaptive neoadjuvant trial in triple negative breast cancer, has really been a model. There are certain advantages in the treatment of breast cancer just by the nature of it being amenable to easily being biopsy pre-treatment and on treatment. But this study now, I think, is up to a total of 9 different neoadjuvant regimens which have been explored in this single platform trial, which has been going on for 10 years or more. So trials like this, and ideally independent trials, which are led by academic groups, perhaps with collaboration with industry colleagues, would be very helpful, I think, particularly where different agents coming from different companies could be brought into the trial in the same way they are in I-SPY2. And of course here, PCR is the primary endpoint. So PCR leads to graduation of a regimen from this trial to a phase 3. And we have the advantage in lung cancer of having that option now as well because of all the data. Dr. Tina Casconi led the NeoCOAST trial, which was one of the first attempts really to look at this. And luckily it happened prior to the chemo IO neoadjuvant era. This randomized patients to one of 3 investigational arms or a control arm of durvalumab single agent just for 1 dose prior to treatment. So window of opportunity, primary endpoint NPR, no post-operative treatment. And these were the results. So not statistically powered really to show differences between the combinations, but visually you can see here that most of those combination arms appear to be leading to more pathologic regression than the durvalumab monotherapy arm. And she has also led the development of NeoCOAST-2. So this is more of a standard of care type treatment. So essentially adding on to chemotherapy plus durvalumab in the neoadjuvant setting bringing in multiple novel agents. So here, for example, you see oleclumab's CD73, monalizumab, which is an NKG2A, and MEDI5752, which is a PD-1 CTLA4 bispecific. So this, the difference here, for example, versus Hudson is there isn't that marker selection. So we're not selecting the patient based on biomarker, which would be the ideal situation, I think, if we had good biomarkers. But on the other hand, we are getting some comparison between the regimens, and we have an external control from the Aegean trial. So how can we make some further advances? Well, these are some thoughts, I think. So if you look at the top here, developing an IO-based agent, and I know these are loose terms, so almost anything people can propose is IO, but you enroll in the study. Ideally, if you have a biomarker, which is some validation for that immuno-oncology agent, then you would select a cohort and perhaps have a third cohort here, which is biomarker selected, a non-selected cohort as well, and a control arm. And that control could be within the trial, or it could be an external control if you have a very reliable PCR rate. Then you get your neoadjuvant treatment. At the time of surgery, you assess the pathologic complete response. There is the potential, I think, in the longer term for accelerated approvals from phase 2 and 3 trials based on PCR. And in the post-operative setting, you could also envisage the scenario of re-randomizing the patients. So depending on their pathologic response, no PCR, randomized to further treatment or no treatment potentially. No PCR, further treatment with PD-1, PD-L1 alone, or a novel combination. Or patients who have a PCR randomized to best supportive care or further treatment, be it as a single agent or combination. On the other hand, we may have to take a different approach to an agent that's purely and really doesn't appear to have an immuno-oncology basis. And these could be things like targeted therapies, for example, KRAS inhibitors. And here it may be challenging to incorporate them purely in the neoadjuvant setting. My thought, at least in the near future, is we may need to incorporate these in the adjuvant setting, perhaps stratifying by PCR status. There's also the idea that perhaps it's not so important to have a targeted agent in the neoadjuvant setting. You're not leveraging that in situ tumor. But this is one other design here in the bottom right. So finally, these are my thoughts. I think pathologic response is going to be a real boon to developing drugs in early stage lung cancer. It should, similar to what we've seen in lung cancer or in breast cancer, it should lend itself well to rapid adaptive designs. However, it's very easy to go wrong with these adaptive designs, as we've seen in advanced lung cancer. And my own feeling is that biomarker guided trials are probably best in this setting, albeit we have particular challenges in early stage lung cancer in turning around results for biomarkers in a very limited period of time. So working very closely as a multidisciplinary team is going to be important. So thank you very much, and I'm happy to join the discussion later.
Sorry, you want to do the questions now? Okay. So we will take questions now for Dr. Ford, since he's still online with us. Are there any questions in the crowd? None for right now. I had you.
So thank you for that lovely talk. I guess one of the things I'm struggling with is what your final conclusion is. Of all the trials and everything you've shown us, what do you think is going to win?
Were you able to hear that, Patrick?
I think the Hudson. Yeah, I was. Can you hear me?
Yeah.
Yeah. Uh-oh.
Can you hear me?
Yeah, we can hear you. It's
sorry.
just telling us the.
I think something along the lines of the Hudson design would make most sense to me, to be honest, because I think that trial really did what it told us. It didn't really give us perhaps the answers we wanted, in that we would like if every regimen showed effect. But what it did do, from a much smaller number of patients enrolled, that's a maximum of 70 patients per arm, it showed that most of those regimens were not going to work in a phase 3 trial. And that's probably what we want to do before we do more phase 3 trials of 700, 800 patients, combining chemo PD-1 plus a novel agent. We want to have a much smaller studies, which will give us early readouts and say it's not useful to pursue this in a much larger trial. I think what Hudson did, though, was also the biomarker matching was very important. And interestingly enough, the putative biomarkers didn't really enrich for most of those agents. So there wasn't. So you might imagine that a CD73 IHC stain might enrich dramatically for response to anti-CD73. Now there may be some relationship there, but it wasn't dramatic. The same with patients with DNA repair alterations in their tumor. Those also didn't appear to dramatically enrich for response. So that would be my kind of ideal approach, where you have a biomarker test up front, perhaps a broad-based approach, NGS plus IHC, depending on the agent. And then you have a number of agents within the trial that the patient can be randomized to. But there are challenges. I think the biggest single challenge we have in lung cancer is getting our results back quickly enough to actually make that viable in early stage disease, where the surgeon and the patient and the med-onc to a degree is not concerned about delaying surgery or delaying systemic treatment. And that could involve scenarios where you allow the patient to have one cycle of treatment, so one cycle of chemo IO while you're doing your testing, and then you bring in the novel agent on cycle 2. But those are, and that's the best that I can come up with. I don't know if you or anyone in the audience has other ideas.
And the last question really is, do you think PARP inhibitors is rational to test in lung cancer?
The data so far in an unselected population would suggest no. So we do have, there was recently a negative trial with olaparib in advanced disease, and the data from Hudson, while there was perhaps some signal, it was not dramatic. There was more data, more supportive data with ATR inhibitors. So I don't think it would make a lot of sense to move them into early stage disease at this point.
Okay, thank you. And thank you for a great talk.
No problem. Thank you.